beam transmits through the sample. Okay, so sample whenever is mounted under the transmission electron microscope, all the inside features of the sample are visible. Imagine an animal cell, okay, a spherical animal cell. So when you mount that animal cell under the transmission electron microscope, how to mount that we will discuss later. Okay, just imagine that you have mounted a spherical animal cell under the TEM. Okay, so all the components inside that cell, all the smaller particles, okay, nucleus or other components of the cell, okay, which are suspended in the fluid inside the cell, will be visible by the transmission electron microscope, because the electron beam, electron rays are transmitting through the sample, okay, and coming out from other side. And that is why we are observing. Okay, so the source in electron microscopy, source is above. In optical microscopy, the source of light is below here. So in electron microscopy, the electron beam is above the microscope. So from above, electron gun shoots the electron, okay, that travels downwards. बीच में sample रहता है. It goes through the sample, transmits through the sample, okay, and comes down, comes out from below. And below we have a set of lenses through which we observe. Okay, so that is how the transmission electron microscopy works. When it comes to scanning electron, as the name suggests, scanning is just for observation of surface features. Okay, so it will, electron beam actually scans the surface of the specimen, of the object sample that you have mounted on the surface. Okay, so scanning is for surfaces, transmission is for in-depth structure observation. Itna difference समझा है? Okay, now there is much more. There are many more complexities when it comes to arrangement of these microscopes. But just as of now, understand the difference between these two. Transmission is to observe what is within the particle, and scanning is to observe the surface structure of the particle. Both the things are very equally important. Okay. Now, observe this image on the slide. Okay, as you can see in the diagram, there is one electron source at the top. Okay, normally there is an electron gun kept at the top, and then there is a set of condenser lenses. Okay, those vary in size. So whatever electron beam is coming out from the electron source gets converged and directed towards the sample, which is kept in between specimen. It is labeled specimen. Okay, and it gets converged and transmits through the specimen. Okay, and when it comes out below the specimen, you have another set of condenser lenses, which again helps us or helps electron beam to get converged and form the image on the plate, which is kept at the bottom. Okay, now the arrangement we will learn more in depth while we are learning about sample preparation or image, how to obtain the image in the microscope. For now, just understand this: electron source ऊपर रहता है. At the top you have electron source, and below that you have a set of condenser lenses, and बीच में in between those lenses you mount your sample. Okay. Now, what is it written here? Because of the very short wavelength of illumination electrons on which the TEM operates, it can resolve objects as close as 1 nanometer. The resolution limit is so very precise, and magnify microbes up to 5 lakh times magnification. Okay, whereas with normal light field microscope, what's normal lab scale microscope, what we can obtain is up to 1000x. So you just for this I asked you, if you have an idea how a 400 times magnified image looks like. Okay, so you can compare if 400 times magnified image looks that clear, that good, then you can just imagine, or we just cannot imagine, how a 5 lakh times magnified image would look. Okay, it looks great. It really looks great. Okay, so let's understand this further. To prepare a specimen, this is very important as compared to when it comes to TEM and SEM. Sample preparation and obtaining the image are two very important points when it comes to writing an answer for these types of questions. We will understand this with the help of this diagram. Okay, it uses the principle of freeze etching or freeze fracture phenomenon. Okay, so what they do, they first put the sample, for example, and just for the sake of example, I'm giving you an example of an animal cell. Okay, so suppose you have an animal cell that you want to observe under the microscope. So they will put that sample, animal cell, inside a block. Now that block can be of anything. It can be a metallic block, it can be a plastic block, or it can be an ice cube. Okay, so ice cube is much more convenient to use, but after ice cube, plastic block is also most widely used. Okay, so they put the cell inside the block, or they prepare a casting around that cell with the help of metal or plastic or ice. Okay, so that your cell is now inside that block. If it is in the ice block, it is in the frozen state. Okay, now once that block is created, you cut that block, you take a section of that block. Okay, imagine the block in the form of a cube. You have a cube of ice, inside which you have placed your cell. Okay, now you are going to cut that cube of ice with the help of a very sharp knife. That knife is made up of diamond. Okay, so you can imagine how sharp it is. So it has to be very sharp so that you can cut through such microstructure. Okay, so with the help of very sharp knife, you section that block. Okay, you take a section of that block, and when you take the section, what happens? That cell which is placed inside in the frozen state, because of section, it now gets divided into two parts. Okay, you cut open the cell by cutting open the ice block, and while cutting open, the parts will get divided. Okay, it will not be a sharp, like plain cut. Okay, you are going to open that block in such a way that the organelles inside the cell will get exposed on the surface like this, like it is shown in the second picture of the slide. Okay, details of fracture of the fractured membrane. Okay, so you are cutting open the membrane basically. So sorry, and that is how the organelles will get exposed on the surface. Okay, after this, you are going to do the last step, which is known as etching. Okay, etching is simply casting those exposed surfaces by certain materials which will be visible under the electron beam. Okay, so in the third image, what you can see, the etched surface of that cytoplasm. The cytoplasm which was exposed by cutting, you have now etched that surface. Okay, so that every projection will be clearly visible on the surface. Now this etched part of the cut open cell to mount on a wire grid. Okay, so on optical microscope you have a plain glass slide on which you mount your sample. In electron microscopy you have a wire grid. Okay, so that electrons can travel directly through the sample. Okay, so you put this etched sample on that tiny, very small copper wire grid. Okay, and that wire grid you mount under the microscope. Okay, so that through that wire grid, electron beam will travel, will come down, will pass through the sample, and will create an image. Okay, which will be observed with the help of lens. Okay, so formation of image, if you can see the next slide, the image that is seen, okay, in the picture is the image of Microcystis aeruginosa. It is a spherical organism. Okay, you can imagine now the organism was put inside the block, in the ice block. That ice block was then cut open from the center. Okay, the upper part was separated, and whatever was left on the surface was etched with suitable material. So what you can, these smaller globule-like structures that you can see in this sphere are the gas vacuoles that this organism carries. Okay, so you can clearly see few projections are concave, few projections are convex. So convex projections still carry gas within those. Okay, and concave projections were cut open and gas was released. Obviously, that is why there is only cavity left on the surface. Okay, so this is how the sample preparation goes like. Okay, now sample prepare हुआ, आपने mount किया microscope में, but अभी you have to observe the image. Whatever you have mounted, you have to observe that through the microscope. That applies, for that you have to apply another set of procedures, because electron beam, electron rays, it cannot directly enter our eyes.